Hello, my name's Julia Edgar and I'm based at the University of Glasgow in Scotland in the United Kingdom. I'm going to tell you today about our work in neural stem cells and how they are able to restore myelin in a mouse model of classical Pelizaeus-Merzbacher disease. I'm delighted to be here and thanks very much to the organisers for inviting me to take part. So as you know, Pelizaeus-Merzbacher disease is due to mutation in the proteolipid protein 1 gene. This disease spans a broad clinical spectrum from hereditary spastic paraplegia type 2 at one end of the scale through classical Pelizaeus-Merzbacher disease to conatal forms of the disease at the other end of the clinical severity spectrum. Clinical severity correlates largely with the amount of myelin that is made and or maintained. And the same is true in animal models harbouring spontaneous mutations in the proteolipid protein 1 gene. So here I'm showing you an image of a brain of a shaking puppy dog. In the inset, you can see myelin in white. That is a section of a brain from a normal healthy dog, and that allows you to compare how the brain of the shaking puppy dog appears with respect to the lack of myelin. I want to point out that this is not an experimental animal. This dog was presented at the University of Glasgow Veterinary School, and it has since been recognised that it harbours also a mutation in the proteolipid protein 1 gene. Anyway, as I mentioned earlier, the subject of this presentation is classical Pelizaeus-Merzbacher disease, which is characterised by a kind of intermediate level of myelin formation and or maintenance and intermediate severity in terms of clinical signs and symptoms. So we recently described MRI and clinical findings in 2 patients with classical forms of Pelizaeus-Merzbacher disease due to proteolipid protein 1 duplication. These were brothers, and they are being examined here by Jim Garburn when he worked at Wayne State University. And as you can see from the picture, the patient has very little muscle remaining in the lower limbs, and that is simply because the central nervous system axons, so the nerve system's electrical wires, if you like, are unable to connect with those in the peripheral nervous system, leading to muscle wastage, loss of muscle tone, loss of strength, and so on. The patient and his brother were wheelchair bound at the time they passed away in their late 40s and early 50s. So we were able to examine central nervous system tissue from these patients. And what we found is that they had actually formed and maintained a considerable amount of myelin. So the spinal cord here on the left belongs to one of our patients. And you can see that in comparison to the spinal cord on the right, which is from a healthy individual, there is a definite reduction in the amount of the brown stain that is present, particularly where the arrows are pointing. And this represents a deficit in myelin in these regions. What we were also able to show is that the myelin that is formed in these patients is rather unstable. So although a considerable amount of myelin forms, this is in contrast to conatal Pelizaeus-Merzbacher disease, where very little myelin forms in the first place. So in these patients with classical Pelizaeus-Merzbacher disease, a considerable amount of myelin formed, but it was rather unstable and prone to breaking down. So as you will know, direct transplantation of neural stem cells has undergone clinical safety trials in 4 patients with conatal forms of Pelizaeus-Merzbacher disease. And we wondered whether this approach might also be suitable for patients with classical forms of Pelizaeus-Merzbacher disease. But bearing in mind that patients with conatal forms of the disease produce virtually no myelin in the first place, whereas, as I've just told you, our patients produce rather a large amount of myelin. So the problem here is that the neural stem cells may not be able to compete with the endogenous or the patient's own cells in order to form myelin around those wires, those electrical wires, or the axons of the central nervous system. So to address this question, we turn to a mouse model of classical Pelizaeus-Merzbacher disease. And importantly, as with our patients, these animals formed rather normal amounts of myelin initially. And that's shown in the black and white images on the left-hand side of your screen. So P40 indicates the age of the animal, postnatal day 40, young adulthood. And in both the image on the left and on the right, you can probably see there is a pale white staining. That is myelin. In the image below, P20, so postnatal day 120, you can see in the left-hand image that a lot of myelin is still present, but in the image on the right of the Pelizaeus-Merzbacher disease mouse model, most of the myelin has disappeared. And this is shown also in graphical form in the graph beneath. So the orange bars represent what happens in the control animal. The amount of myelin present in the brain increases over time, whereas in the PMD model, the amount of myelin gradually decreases. The picture on the right is of Frederick Grinnerfelder, who was my PhD student who did this work. So just to summarise so far, what we have shown you in both the animal model and in patients, oligodendrocyte precursor cells, indicated by the cell on the left, are able to form myelin, so the cell in the middle there, labelled OL. They're able to form mature myelinating oligodendrocytes that wrap axons with myelin sheaths, but that myelin is rather unstable and subsequently breaks down. So if you move to the right-hand side image, there the oligodendrocyte has lost its myelin sheaths, and there is infiltration into the central nervous system also of immune cells that are not normally present there. And these immune cells produce inflammatory factors that can actually harm the myelinating cells and the neurons that they wrap. So, if neural stem cell transplantation is going to help patients with classical Pelizaeus-Merzbacher disease, it is going to have to restore stable myelin and to prevent neuroinflammation. To test if neural stem cell transplantation might represent an appropriate therapeutic approach, we took neural precursor cells and injected them into our newborn PMD mouse model. We waited for 14, 35 and 120 days, represented in the images on the top there, 14 days post-transplantation, 35 days post-transplantation and so on, and examined the brains of the transplanted mice. And what you can see indicated by the dark material in these upper three images is that myelin has formed from the transplanted cells. In the images on the very bottom, and these are labelled electron micrographs, these are images taken using a very high-powered electron microscope, and they look at the very ultra-structural detail of the transplant-derived myelin. And we can see in the two right-hand images the axon overlaid in purple in the centre, surrounded by a thick and healthy myelin sheath. So this was really good news for us. This really suggested that despite that these animals can produce PMD myelin, if you transplant neural stem cells into these animals at an early age, then these neural stem cells can go in and outcompete the endogenous or PMD cells and form normal, healthy myelin. A question remained, however, and that was, do the neural stem cells also prevent neuroinflammation? So to assess this, we used macrophages, so the cells shown in purple in the top three images, as a measure of neuroinflammation. So macrophages produce inflammatory factors. So we examined the tissue of the non-transplanted and the transplanted animals and quantified numbers of macrophages, and we found that there was a significant reduction in the number of these cells in the transplanted animals, really indicating that cell transplantation of neural stem cells not only restores normal healthy myelin, but also reduces neuroinflammation. So finally, just to summarise all that, in the PMD model and in patients, oligodendrocyte precursor cells can produce myelin, but that myelin is somewhat unstable, it gradually degrades, and cells of the immune system enter the central nervous system, producing inflammatory factors and contributing to damage. If we transplant healthy neural precursor cells, shown here in green, they can outcompete the PMD OPCs, oligodendrocyte precursor cells, shown in blue, to form stable myelin sheaths. Neuroinflammation is also reduced. And finally, I just want to thank my colleagues in Glasgow and our collaborators elsewhere in the world who were involved in this project, alongside the funders. And most importantly, I want to say a very special thank you to Jack and Gary's mum and their brother, who allowed us to use the tissue samples from these two patients, and who I'm very, very happy to remember here in these pictures that Jack and Gary's brother sent me.